Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Have you ever just stopped and wondered, what's actually happening, like deep down at the tiny level when you learn something new? Or maybe when a memory just pops into your head?
Right, it's all about the connections, isn't it, between your brain cells, the neurons.
Exactly. But what is it that keeps those connections, well, strong and flexible? How do they adapt?
That's one of the big questions in neuroscience, really. Our brains are just incredibly dynamic. And yeah, that ability we have to learn, to remember things, it hinges completely on how those neurons talk to each other and, crucially, rewire themselves.
And that brings us to our deep dive today. We're looking into a really groundbreaking discovery focusing on this fascinating brain protein. It's called Cypin.
That's C-Y-P-I-N.
Right, Cypin. And look, this isn't just, another protein on the list. It seemed to be a key player, almost like a sculptor, maintaining those really important communication lines between neurons.
Lines that are absolutely vital for things we rely on every second, like learning and memory.
So our mission here is to unpack how Cypin actually works. Its mechanisms are, well, pretty surprising. We want to reveal this role it plays that wasn't known before.
And explore what this all means, the implications, especially for understanding and maybe eventually treating things like traumatic brain injury or neurodegenerative diseases. We're drawing on a recent scientific paper and a related health news piece for this.
Okay, let's get into it. This hidden sculptor, Cypin. When we talk about learning, about memory, we're talking about the brain changing fast, really fast.
And those changes mostly happen at the synapses, those tiny little gaps where one brain cell passes a message to the next.
And this whole process, it needs really quick, precise adjustments to the proteins that are right there at those connection points.
That's spot on. And a really common way the brain does this, makes these super fast changes, is through a process called ubiquitination.
Ubiquitination.
Yeah, you could think of it like attaching little tags, almost like sticky notes, onto proteins. And these tags, they're instructions.
Instructions for what?
Well, they might tell the protein, okay, move over here, or turn on this function, or even time to be recycled.
Ah, okay. And this is where it gets really interesting because apparently not all these tags are the same, are they? The study looked at two main types of, polyubiquitin linkages.
Exactly. Two main types they focused on. The first one is known as K48 polyubiquitin.
K48?
K48. And historically, these K48 tags were mostly understood as signals for basically destruction. They mark a protein to be sent off to the proteasome.
The proteasome, that's the cell's recycling plant, right?
Precisely. It breaks down old or damaged stuff the cell doesn't need anymore. So K48 often means get rid of this.
But the big news here, the real kind of revelation in this research, is about the second type, K63 polyubiquitin.
Yes, K63. And unlike K48, these tags seem less about destruction and much more about, well, organizing things, building things, controlling signals.
Okay, so not recycling, but something else.
Right. People have studied K63 tags in other contexts, like cancer research, where they play a role in signal transduction, basically how cells get messages and respond. But what they did specifically in neurons, right at the synapse, that was largely a mystery.
Until now.
Until now. And this is where Cypin comes in. The researchers found that Cypin is a crucial regulator of proteins linked with these K63 tags, specifically in neurons and right there at the synapse.
So Cypin is managing these K63 tags.
It seems to be. And the way it interacts with that recycling plant, the proteasome, is really unexpected.
What did they find?
They found Cypin actually grabs onto a specific part of the proteasome, and by doing that, it actually slows it down. It inhibits its activity.
So it stops the recycling.
Well, it slows it down for certain proteins. It leads to a buildup of ubiquitinated proteins, especially, it seems, those carrying the K63 tags. It's almost like Cypin is saying, whoa, hold on, don't recycle that one just yet. We need it.
Okay, that's fascinating. So it's not just about adding tags, but also managing the removal process.
It looks that way, and they saw this directly in neurons. When they increased Cypin levels, the amount of proteins with those K48 recycle me tags went down.
Decreased. Okay.
But at the same time, the levels of proteins with the K63 tags went up.
Ah, so it's specifically promoting the K63 type.
That's what the evidence strongly suggests. Cypin seems to have this unique role in favoring the K63 tagging pathway, which, remember, is more about stability and function, not breakdown.
So how does it do that? How does Cypin actually boosts these K63 tags? Did they figure that out?
They did find a key piece of the puzzle. When Cypin levels went up, it also significantly increased the levels of another protein. This one's called ubiquitination factor E4A.
Ubiquitination factor E4A.
Or UBE4A for short, but let's stick with the full name. And this factor, ubiquitination factor E4A, is already known to be an enzyme that promotes adding those K63 linkages.
I see. So Cypin boosts the enzyme that puts on the K63 tags. It's like hiring more workers for a specific job.
That's a great analogy. And the effect this has on the synapse itself, on its building blocks, is pretty profound.
What kind of effect?
The research showed that more Cypin meant higher levels of really crucial synaptic proteins. Things like scaffolding proteins, the ones that physically hold the synapse together.
Like the structure itself.
Exactly. And also key receptors, like specific types of NMDA and AMPA receptors, which are absolutely vital for receiving signals from other neurons.
So it's beefing up the receiving equipment.
It looks like it. These findings strongly suggest Cypin enhances how receptors work and fine-tunes the way signals get transmitted across the synapse, making sure everything's in place and ready.
And what's really striking, you mentioned earlier, is that this isn't just happening on one side of the connection.
No, that's a key point. It affects both the presynaptic side, that's the neuron sending the signal, and the postsynaptic side, the receiver.
Both ends of the conversation.
Right. And interestingly, when they increased Cypin, the biggest impact they saw seemed to be on the presynaptic side. Things like how the synapse is organized, how materials get transported there. And again, this seems to happen via that K63 polyubiquitination pathway.
So it's optimizing the whole communication setup, really,
Yeah.
from sending to receiving.
That's what the data points towards. And it's worth mentioning, these weren't just findings from cells in a dish. They did studies using developing neurons in culture. That's the in vitro part. But they also confirmed key findings in adult mice in vivo, meaning in living organisms.
Okay, so seeing it both in isolated cells and a whole living system adds a lot of weight, doesn't it?
It really does. That consistency gives you much higher confidence that this is biologically relevant, that it's actually happening in complex brains.
So let's step back. We've got this intricate dance of proteins, Cypin managing these K63 tags, influencing the proteasome. Why does this matter to, us,
Yeah.
to our listeners?
Well, first off, it directly impacts learning and memory. By ensuring the right proteins are present and stable at the synapse, Cypin helps neurons communicate effectively.
Stronger connections, better communication.
Exactly. Which means Cypin is helping maintain those brain cell connections that you absolutely need to learn new skills or recall information.
And this relates to that idea of synaptic plasticity, right? The brain's ability to change connections.
Absolutely. Cypin seems to be a significant player in promoting synaptic plasticity. That incredible ability of the brain to strengthen connections or sometimes weaken them based on experience.
Which is fundamental. That's how we learn, how we adapt.
It is. Without plasticity, learning as we know it just wouldn't happen.
And thinking bigger picture, the potential for therapies here seems huge.
It really does. Think about conditions where synaptic function goes wrong. Neurodegenerative diseases like Alzheimer's or Parkinson's, or traumatic brain injury.
Where those connections are damaged or lost.
Precisely. And Bonnie Firestein, she's a distinguished professor at Rutgers and one of the authors on the study, she pointed this out very clearly.
What did she say?
Well, she noted that developing treatments that specifically target Cypin might help improve those connections between brain cells, which could, in turn, enhance memory and thinking abilities. She added, and this is almost a direct quote, that these findings suggest Cypin could be a target for developing treatments for neurodegenerative diseases, neurocognitive disorders, and brain injuries, too.
That's a pretty strong statement.
It is, and it's backed up by other observations, too. For instance, Cypin levels are known to go up naturally after a traumatic brain injury.
As if the brain is trying to use it to repair itself.
Perhaps. And other studies have even shown that activating Cypin can help restore certain types of memory, like fear conditioning, after it's been disrupted. So the potential seems really tangible.
Okay, but like all research, there must be limitations or next steps.
Right.
Where does this go now?
For sure. Good science always asks more questions. The researchers themselves pointed out a few things. For example, while those neuron culture studies are powerful, they can't fully mimic the sheer complexity of a living, intact brain.
The connections are just simpler in a dish.
Right. But again, the fact that they saw consistent results between the in vitro work and the in vivo studies in mice is really encouraging. It suggests the core findings hold up.
What else?
Well, just studying ubiquitination itself is technically very challenging.
Why is that?
Because these tags, they can be put on and taken off very quickly. They're transient. And also developing the tools like specific antibodies to detect every single type of ubiquitin linkage reliably is difficult.
So it's hard to get a perfect snapshot of what's happening with all the tags all the time.
Exactly. And one more thing, most of the experiments in this particular study involved increasing Cypin levels.
Which you said mimics what happens during activity or injury.
Right. But future work needs to look at the whole picture. What happens if Cypin levels are reduced? What's the full dynamic range? And how do these different ubiquitin tags, K48 and K63, maybe even others, interact? It's complicated.
A lot more to explore then.
Definitely. It's a rich area.
So we try to boil this down. What's the key takeaway here?
I think the big takeaway is that Cypin emerges as this really sophisticated manager inside the synapse. It's primarily using its ability to promote these K63 tags, which are about function and stability, while also having this unexpected influence on the proteasome, the recycling system.
The sculptor analogy seems pretty apt then. Ensuring the right pieces are there, shaped correctly for communication, learning, memory.
I think so. It's ensuring the synapse has the right building blocks properly configured.
In this work, it's a great reminder of how important that basic research is, isn't it?
Yeah.
Understanding these fundamental processes.
Absolutely. Like Bonnie Firestein emphasized, you have to understand the basic mechanisms first. That's the path that eventually leads to practical treatments down the road.
So a question for you listening. What really stands out to you about this idea? That just one protein can have such a complex, multi-layered impact on something as massive as brain function.
Yeah, it's pretty mind-bending. And maybe a final thought to leave you with. Consider that these subtle tags, like the K63 one Cypin promotes, they aren't about destroying things, but about signaling, organizing.
A different kind of protein language.
Exactly. Maybe it's a kind of silent language happening constantly inside our neurons, shaping our thoughts, our memories, our intelligence in ways we're really only just starting to decode.
Makes you wonder what other hidden signals are still out there, orchestrating things inside our heads, just waiting to be discovered.
It's an exciting frontier.
Well, that brings us to the end of this deep dive. Thank you so much for joining us.
Until next time, keep exploring the incredible world inside you and all around you.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.